I'm going to be speaking briefly about natural history studies and what it is that we need to do for the GABA-A community and what it would take to get centers of excellence started and what we need to understand about natural histories in order to progress treatments. So to start with, why are natural histories important? First of all, it helps us to understand what the disease is like. It helps to identify symptoms beyond the obvious ones that the disease or variant might initially have been associated with. So here it's epilepsy and seizures, but what other symptoms are maybe very prevalent in patients and just as important to their quality of life. It also helps to determine questions about how the disease progresses through a lifespan. If, for instance, there is one sort of seizure present in infants and then it progresses to a different type of seizure later in life, or if people outgrow seizures as they age, or any other factors that might be changing as a person ages. Natural histories can also help us understand modifying factors. This could be modifying genes, or it might be things such as other illnesses that impact the way the disease manifests, such as if a child catches the flu and their seizures get worse for a while, or if they experience regression. Or these factors could also include other medications and supplements they might be taking and how these impact the GABA-A variant manifestation. Natural history studies can also help to determine clinical trial endpoints. It lets you know which symptoms are most common and which are of most concern to patients, which are the most severe symptoms, and also, as I mentioned before, age-specific symptoms. Because if you have a type of seizure or any other type of symptom that is only present in a specific age group, that would tell you a lot of information about how to be designing clinical trials, what ages of enrollment, what inclusion and exclusion criteria that you might need. If you don't have this information, your clinical trial may fail just because your patient population didn't actually end up having the type of symptom that you thought you were going to be treating, or they outgrew it naturally and it wasn't due to the intervention. And the natural history studies they have the potential to replace placebo control groups for rare and devastating disorders. If the natural history is understood well enough where there's a solid understanding of what a person with a specific gene will normally look like, what the progression is, then possibly a placebo control group can be replaced with by just comparing patients receiving treatment to the known progression of the disease. There are quite a number of natural history platforms out there, and they fall into two broad categories. One is those that compile a patient's electronic health record across the various hospitals and institutions they may have been to, which for many of these patients is quite a number of different hospitals, and it is difficult for parents to keep track of all that information or for any researcher to assemble all of that themselves. So platforms like Citizen and All Stripes can collect all of that information in one place and make it available to researchers. There are also other natural history platforms that are patient or caregiver entered. So these will use sometimes standardized questionnaires and surveys such as the Vineland or quality of life assessments, but they can also include information that is very customized and individual to what matters to that particular patient. So that could be something about, whether it's toileting abilities, and you can see how a patient is doing with that, or sleep quality, or, just their communication abilities in ways that maybe don't get captured on regular assessments as much. But these patient-entered platforms allow for those unique bits of data to be collected and gathered. And then the Cure GABA-A variants group is beginning some natural history studies. So this foundation is brand new. We've only started this year, but already there's a little bit underway. We are using the Citizen platform. There are 6 patients currently enrolled, but we plan to get many more next year. And then Matrix is the caregiver-entered platform of choice for Cure GABA-A, and that will be launching at the beginning of next year. And I just want to mention that in conjunction with the natural history data that is on Matrix, this will also be tied to samples being collected in a biorepository. So you will have extensive phenotypic data to go with all of those different patient-derived samples. Right, and with that, I would like to open things up to a discussion for the next around 20 to 30 minutes about what it would take to develop more formal natural history studies and centers of excellence that could potentially be used as clinical trial locations in the future, and anything else that we need to understand about the clinical presentation of patients that would enable us to reach cures faster.
Since I'm situated right next to you, and I'm Phil Pearl from Boston Children's, I have a strong delegation behind me who will correct what I'm going to say. But one thing I want to point out that we've learned the hard way, sort of, in our natural history study. We're conducting a natural history study right now of succidic semialdehyde dehydrogenase deficiency. So it's a GABA disorder where they can't break down GABA. They have tremendously high GABA levels. And some of our disease controls have GABA transaminase deficiency. They have even higher GABA levels. But the point I'm trying to make is these are brains in development. So there are compensatory changes that change everything. And we have learned so much from doing a natural history study. It's internationally based. We have one U.S. site, that's Boston Children's, but we are working with the International Neurotransmitter Disease Association based in Heidelberg, and we have other European sites. And we also have standard of care sites. This is important. That's for patients who can't get to a center, but you can still collect their information. And we can't do all the fancy studies that we're doing, like MR spectroscopy with special GABA editing using megapress technology, which is how you can quantify GABA, and MEG studies with Christos Papadopoulos next to me, and high-density EEGs, and finding out abnormalities in the linearity index that an induced potential is affected by GABA. But the point is, we've learned over time, for example, in these patients, that the GABA levels just change dramatically over time because their brain's compensating and they're downregulating their GABA-A receptors, which are bombarded, so they downregulate, and the GABA levels, GHB levels, the other metabolite levels change. So the point I'm trying to make is in these developmental, as Mike Rogowski said, these are not just epileptic encephalopathies, these are kids going through development. So in these DEEs are all these compensatory changes that have really affected the way we have to look at the disease. In addition, epilepsy can be progressive. So we've learned that the SUDEP rates are exceedingly high in the adolescents and adults. So the epilepsy, and by the way, this is a syndromic form of autism. Most autism, as a child neurologist, you can pretty much diagnose it by age 4 or 5, if not earlier. But these kids aren't showing autism until ages 7 to 10, and the epilepsy typically has onset at the same age, which is when the GABA levels drop. So it's interesting. You lose the GABA levels and the epilepsy starts up, and then the SUDEP rate gets to be about 15% in adolescents and adults, whereas we're not seeing SUDEP in our pediatric patients. So I'm just trying to make the point we're dealing with the developing brain.
Thank you very much. Not going to try to pass the microphone around because it's tethered.
It's not so much to correct anything that Phil said, it's to complement it. So just a couple of one somewhat of a caricature way to look at any mutation that causes an epilepsy that we wish to correct for purposes of a cure is there's roughly 3 levels of abnormalities to correct. One is the primary mutation to replace whatever might be missing. The second one, as Phil said, is compensatory change. Many times for the organism to survive, something has to be upregulated or downregulated in compensation. If a patient is missing enough alpha-1 subunit, some other subunit may be overexpressed in compensation. Or if a patient has an excess of synaptic GABA, there may be downregulation of synaptic GABA-A receptor. So the second one is these compensatory changes that are there in utero, usually, and pretty early in life probably, just for the organism to make it, to breathe. The third is something that we as epileptologists may not be paying enough attention to it, are changes that are there in epileptogenesis. As Phil said, these are progressive syndromes. So the third layer of abnormalities, especially in the encephalopathies that come with epilepsy, are the consequences of recurrent seizures. What happens to the brain at the neuron level, at the astrocyte level, after it's had a few rounds of recurrent seizures? Some of these seizures are conspicuously injurious. You can see it on MRI. They will come with the changes that will sometimes follow head trauma or infection or something else that will eventually cause epilepsy. It's not entirely clear that those kinds of changes won't have to be unwound separately from fixing the primary problem. So in other words, there's an interesting question of what happens when you fix the primary problem. Will the compensatory changes jump back to normal? In some cases, if they don't, that might be a problem for the patient. For example, in a disorder of excess synaptic GABA, if you restore the enzyme in question that's missing and normalize synaptic GABA, but the dynamic of the restoration of the receptor is not at the same pace as the restoration of the neurotransmitter, you might expose the patient to seizure. That may require, in thinking about how we approach therapies, in co-administration of a gene and a drug, at least for that window when you have to unwind the compensatory changes. The other level of change, what happened to the brain after hundreds of seizures, may or may not need a second drug. In other words, those changes are that of an acquired epilepsy. And those may or may not bounce back after you fix the primary genetic problem. Or if they do bounce back, it may be a year. And there's a hint of that in the recent Stoke therapeutic data that we saw yesterday at the SLC meeting. I don't know if anybody's here from Stoke, but with all due respect, the data were modest last year, but are conspicuously better this year, after triple-digit number of days have passed since administration and at a good dose of their product. So that's just, I think, a rough and, again, a caricature way to think about the categories of change that we want in the patients whom we want to cure. We want to replace the thing that's missing. We want to accommodate the compensatory changes, maybe by bridging with a drug while those compensatory changes bounce back to normal. And then for those patients who have an epilepsy, be mindful of elements of an acquired epilepsy that are probably there on top of the congenital problem that they have. Now, I hope that makes sense.
Yeah, I can speak something. Yeah, so because we study GABA-A receptor as the documented down, and that have regards to talk about the documented down and being worked for over 50 back next 5 decades. So we know that at least the 10 subunits are associated with drug. We can find the efficacy. So for natural history, I can connect by phenotype or by gene. That would be very important. Also, the some gene plays up alpha 3, beta 3, we know is very rich in development that I'm ready with it. But later on, the plateau and the beta 2 will be upregulated. Career postdoc, he actually record defects in the hippocampus. So the change is very dynamic. So by symptom or by gene, or natural history, that will can think.
Sir, this is fascinating discussion, and I don't want to derail it, and I want to turn it back to natural history. And how do we set up centers of excellence? Because what this points out to me is how badly we need these natural history studies and how badly we need these centers of excellence. And what you showed up there, Matrix Citizen, that can actually be a way for a lot of centers of excellence to pool data in one place. I want to make a plug right up off the bat for however many of you want to have a natural history site, please do so, and please pull your data in a central repository. These variants are too rare to be studied in individual silos.
Thank you very much for that, Gary Joe.
Just jumping on that, that's our experience as well. The more the merrier. It's really powerful when you start getting the numbers up to the triple figures, for instance, which you may or may not be able to achieve, depending on whether you're looking at individual genes or not. I'd also say our experience and with natural history is to have a little bit of a think about what phenotyping you're going to do as well. I think that's really important. So we've seen issues where, as you say, we start with one patient, and that patient might have a particularly severe phenotype, and then make an assumption that that phenotype is for that particular gene. Have another similar gene somebody else is working on, they don't see that phenotype, and so they make an assumption that phenotype isn't there, and they don't ask for it. So they don't collect the data. And then all of a sudden, when you start getting a lot of patients, publications go out that this is important for this gene, but not for this gene. Do a ring around and you find, it's actually important for both. So I think that's, a really important lesson that we learned, that to be thorough about the phenotyping. And the other thing that we've learned is EEGs. EEGs are really important, and they can be difficult to track. So we'll have a lot of patients, and we'll only have a very small, I think they published a paper in 90 years, and I believe we're only able to get 8 EEGs when we just do that we are not enough to look at. Might have been 9. I think we only ended up reading 8. But yeah, like, that's one thing I'll state right from the get-go, that make sure you know where the EEGs are and they don't get lost and get deleted, because I know they're hidden data and they can but yeah, they're really important.
Well, we think that, it's actually Nathan's work, the stratification around functional impact. I mean, that's what Nathan's been doing. Having in SCN1A, 8A, there's gain and there's loss of function. That's probably a gross simplification that already patients are stratifying in that realm. So the heart of this thing has to be fundamental scientists looking at the functional on it, because that's going to help the stratification of these different populations, and that absolutely going to change how these patients are treated. It's true for all the other genes as well. So that is,
Going back to that point and that point about sort of merging everything and having one big data set, sometimes it's not a bad thing if you have 2 data sets. It's a bad thing if you have 7. Doubtless. But having 2 may not be a bad thing. Sometimes there are times where you find something in one data set, and it's really nice to have an independent data set to go and test back against. So when set up the natural history study, and have a center of excellence, I think don't be afraid if there's like a second data set that's independent, a little bit different to yours. That can be just as powerful if everything's merged. And particularly when you have different ages. So you've got to think about what age you want all the data for, and when you're excluding people. So you may sort of be able to say, right, well, we're only taking people from age of 5 and under, for instance. That's great because it gives you another data set. These older patients, they may not have the same data. They may have to chase up a little bit of data, but then, you've got a bigger and a smaller data set to look at, and that can be powerful too.
I'd like to add to the conversation the challenges with regard to having patients with variants of significance as a very common challenge in the clinical sphere, right? So when we test them, we get those that have no pathogenic variants, and then those where the variants, we don't know if it disrupts the function of the gene, the protein function. And so I think that there is value in sort of developing either high throughput or streamlined approach to be able to better characterize some of these variants. And that would certainly increase the number of patients that we have that can participate in the various.
Yeah, that feeds into a thought I had, and this is more of a question rather than a statement. But, the number of electrophysiologists on the planet seems to be shrinking.
Rare breed.
Yeah, agree?
Very rare breed.
Yes, very much so. But there are companies, contract research organizations that use automated patch clamp systems, and they can do this kind of work relatively routinely, I think, in more of a production industrial strength fashion. Now, I don't know how you're planning on funding this initiative, but whatever funding source you go to, I'm asking, would it be possible to write into that a component that contracted with one of these CROs so that you routinely could get a functional analysis of the GABA-A receptors? It seems like a very straightforward approach. Now, there's obviously lots of things that Caddy does in her lab that probably are more specialized in terms of trafficking and so forth. But just to characterize the activity of the GABA-A receptors with the variations to get at your question, is this pathogenic or not, I think would be really worthwhile.
Agree, and to add on, so because Dr. McDonald has worked on it for 5 decades, we have a lot of tools built. So we have developed a high throughput flow cytometry. So in epilepsy field, people don't use flow cytometry, but we have used that about 10 years ago. So basically, we know which antibody is good, which one is good for surface, which one is good for total. So we actually have a pretty good understanding about, for example, if you tell me you have a mutation, this variant in alpha 1, that one in beta 2, beta 3, we can plot pretty quickly about how much at the cell surface. So this will give you some idea about how much overall function you're going to have if you have a mutation alpha-1, for example, a certain variant. Because when we assemble with like a recombinant receptor alpha-1 beta, we usually do beta-2 and gamma-2. These very common isoforms. So you compare with the wet, a very good control, you have a 20% or 30%. So we know in patient condition, they're good in loss function. And also we can plot the difference between heterozygous and homozygous. So in a homozygous condition and the heterozygous, which is patient condition, we can tell which allele is function. So this why we are standing on a giant children. So this is why we already have the tools for that. So yeah.
Let's talk about it.
Yeah, I know. One thing that we learned very quickly when doing high throughput screening of functional variants, and this was work that we did with Steve Tranellis' lab at Emory, was that what was happening in oocytes was not necessarily what was happening at the organism level when we introduced this. That's, CRISPR variants. And a lot of that has to do with the regulatory kind of mechanisms that a neuron has to get rid of misfolded proteins and mutated proteins. And so I would really caution this foundation against these sort of really high throughput initiatives where you might lose a lot of the sensitivity. And especially since the clinicians have observed that a lot of this is loss of function mutations. You may see something that looks like an ADA function, within a frog's egg that doesn't translate. So I would caution against that and look more towards the phenotypes to help kind of drive whether someone is more likely to be categorized as surely someone who has a variant, or whether, we're still uncertain about that. And they still have a PUF.
The point that I think is really important is that it's got to correlate with things. And you make a really good point as well. It's something that we spend a lot of time doing. He's thinking about variance, uncertain significance, and after on. The thing that I think is not done well in academia is the negative controls. It really doesn't matter what your answer is if you don't do your negative controls. So if you don't go and pick variants from the normal population, which don't cause epilepsy, and if you don't have those in your screen, whether you're doing high throughput or not, then you don't know how robust your mission. And so when you start putting those normal controls in, then you get a really nice range of what the normal population that is. There will be plenty of variants that pop into that that we don't know. That's okay. But when we do that, we can start seeing exactly what the range is. So when you're outside in that, this is like that's in that case. And so whether you're doing surface expression, whether you're doing oocytes, whether you're doing HK cells, it's really important to do that. And then they validate the technique. So the technique shouldn't be validated because I think X do this and I think oocytes do that. It might very well be that in some receptors, X are great, myocytes are chill. And might be the other receptors. You'll only know that if you put in your negative controls and you blind yourself to those. So blinding is really critical. You want to be doing the experiments where you don't know, the operator should not know which of those variants are controlled, which one is possible. And that's not done enough, I fear, in a lot of that. So I think when you do that, you have to set up the parameters of you have to set up the parameters to your starting, so it's rigorous, so in your hands.
Yeah, so that is a wonderful point. And I think that's something we might address later in the translational section. But for now, we're trying to keep it focused on the very clinical side. So how would we address the VUSs from a clinical setting and natural history? Would we want to be enrolling people with any variant at all in a GABA-A receptor, and then look for something like EEG biosignatures or a plasma biomarker or something like that to rule out what is just a pathogenic versus benign variant? Thoughts on how we could do that, in the humans, to address what Catherine said?
Yeah, no, I think you're right on, Sarah. And try to figure out what characterizes the average, GABA-A patient. So, you can run studies comparing the 4 serdine kind of diagnosed Crohn's, which is a BUS is a C, who is fitting in with that general category, whether it's average of that category matches your certain variants, and then like look for outlier cases. So to your point, is there a certain signature on the EEG that definitely indicates this is not, a GABA-A receptor including variant? Is there like a certain response to a medication that is a red flag? Looking for those outliers is about important.
Other small diseases are going through the same thing. So, for example, I'm a member of a variant curation group for AADC deficiency, aromatic aminoacid carboxylase. So we're actually doing gene therapy now. And we meet every month. So we've been meeting for 2 years, curating all the variants. So I would say take the model that's out there. On kind of AADC gene variant will at the tertiary results and me. Either they have geneticists who spend all their time discussing these variants based on in silico information and correlating it with the phenotype. Because you're gonna have a lot of benign variants out there. You don't want to be including others in your study. Right. Forget it.
Yeah, I think also, though, it's important to consider that when a disease is first identified, you see the very severe cases, you get kind of the classical image of what it looks like, and we don't want to rule out those who have true mutations in the genes, but it's causing a non-classical presentation, or maybe just a milder, they're not normal, but it's not as severe as you might think. So, we need to be sure to kind of consider both sides of, is it actually GABA, but just in a way that we're not used to seeing GABA receptor in.
I'd like to look at all of there. Well, so sometimes I get a counter from parents. They say, my baby does you already from today. No seizure yet. 2 years later, now I'm the seizure. So maybe the time getting you look more than brain. But I haven't looked before. Well, me just show some sign, some very smart turns in his back. At family. And then, so of course you guys from most kids.
Yeah, even that's the importance of a longitudinal natural history study, absolutely, to figure out what those early warning signs are.
Yes. I wanted to add another perspective about using exogenous expression systems like that.
Can you speak up a bit, please, for that? Sure.
Given the stage of this endeavor and the large number of variants that are going to be discovered, there's a huge need to have a kind of a characterization process that and has a number of standards to it. And I completely agree that there are that you can't directly translate structured function in a zoocyte to what's going on in the brain, but at the same time, you can figure out things like, well, are there basic properties of the receptor that are changed by this mutation, the affinity for GABA or the channel inductance, etc. And having that kind of an objective database is still very helpful, I think. And you can always keep in mind that it is a database that's been acquired from an exogenous expression system, so it's not the gold standard, but nevertheless, I think it can be very useful. It's stratifying patients from it does, yeah. That's what Nathan's showing, that the really early onsets of gain of function, and we've actually made a mouse on the gain of function variant, one of the Nathan ones, and it does have bigger synaptic responses, so it is, the oocyte's not perfect, never going to be perfect. I was a bit skeptical, but it is actually larger, so there is some translation of these simple systems. You can't take them carte blanche, but you can definitely mark you. And I know you're not wanting to do that, but the SEN2A realm, gain of function, it's critical to stratify because otherwise you can't use an ASO-based treatment. You've got to stratify on a functional basis at some level.
Are we able to tell, from the phenotypic presentation, who has a GABA-A gain of function or loss of function, or is that something that
That's what you should figure out.
Okay, so the clinicians agree that's something we do not know yet.
And I think that's critical. A functional relationship with the phenotyping is critical. And this is telling you a lot later on about which therapeutics are going to be beneficial for group Asia populations. I think then critical infrastructures that meets near at the forefront of the center.
Basically, I'm not against using xenophil sites as the initial study system, to carry out the function of the mutant GABA receptors. But as Michael just mentioned, there's really nice technology development these days. For example, many companies, basically the 3 major companies are making automatic patch clamping system. Those patch clamping systems are really, have really nice capacity to study the function of the GABA receptors, right? So basically, and also, so our department purchased one automatic patch clamping system from Frazion. So it's amazing. Within 10 minutes, we will be able to measure a ECP value of the 4 of 8 mutants very quickly. So the data is very robust. I think with this technological advancement, using hex cells for the initial functional characterization is not very difficult. It takes time and takes energy, but it's definitely doable to use the hex cells as the initial functional characterization. And also hex cells and then neurons, but many people think that hex cells have really similar environment compared to neurons, right? If we consider about the current GABA variants, for the major subunits of GABA variants, like alpha 1, beta 2, beta 3, and gamma 2, each of these subunits has over 200 missense mutations being identified in the clinical variant database, right? But very amazingly, over 70% of these variants have no functional characterization. I think probably it's a very important and really I think one of the priorities should be just to a functional characterization of all these variants. Surely now we know there's many of the in silico prediction software that is a recently developed AI derived this alpha missense mutation, which was just published in Science, right? So they have all this, they predict all the saturating mutations of the human proteome. I think that will give us really nice clue about whether mutation either it's a clinical variant or the variant has not been identified clinic. That will give us a really nice clue that whether the mutation can be pathogenic or just benign, and then we can continue to verify those variants using the highest throughput functional assay. I think that's it's doable, and it's just takes time.
Let me just comment on that, because I think that's kind of what I'm imagining, that you'd have an industrial scale CRO that would run through not only the disease-associated variants, but other variants that occur and that we don't really know anything about as controls. And they would be getting these on a coded basis. They wouldn't know anything about the disease, so it would be totally on a blinded fashion. But they would be done, under standard conditions, so you compare from one to the other. The problem that we have is that you have one postdoc doing an experiment on, one set family, and then you've got another lab doing it under different conditions, and you really can't compare. And so the field is unfortunately kind of a mess because of that. If we had kind of a standard procedure, almost like the anti-epileptic drug screening program at the University of Utah, where we run all of the anti-seizure medications through their standardized protocols, and we can kind of compare from, one company to the next because they all send it to Utah. If you look at data that isn't generated in that fashion, it's sometimes really confusing. But having something standardized in that fashion, I think, is really critical.
I had 3 voices that I wanted to bring to the conversation. One is the parent voice. If you have a natural history study, you get to know parents, and the physicians get to hear their stories, and you can collect those stories in a formal way through disease concept models, to qualitative interviews and things like that, to really understand what are the aspects of the disease that are most influential to them. The second is the physician's voice, that there's an intuition. So every time I see Dr. Paul, I'm like, there is nobody who knows more than FSHD deficiency than him. And there's probably aspects of the disease that nobody really understands the nuances as well as Dr. Pearl. And so you want to develop a cohort of physicians that really intimately understand this population. Because the voice that you need to speak to is the regulator's voice. So if there's going to be novel therapeutics or repurposed drugs or whatever, the FDA needs to sign off. And so the reason that's important is that all of the parent's voice, the clinician's voice, is going to help you choose outcome measures in a clinical trial. One of the main benefits of sort of the modern natural history study is that you're collecting sort of it's like FDA quality data, so that you know what your background looks like, you know what the typical trajectories are. So if your drug or your intervention gives you a signal, you have the background that you know is different. And then, when you have that, then for the, I'm hearing lots of strong scientific voices, that clinical intuition then communicates with the variants and with the physiologic phenotypes, and you can start to tease apart if is there type A and type B, or is this a spectrum? And that's also going to help with clinical trial design when therapies are identified for their certain.
Thank you very much. I think we are going to move on to the next section now. This will be more on the basic science. And so we'll have Kieran Normoyle do that.